So for those of you who may not know me, I am Molly Fuente. I am the Assistant Director for the Proposal Development Unit here at NC State, and I lead the Agriculture, Health, and Life Sciences team. So you may actually know my team member, Amanda, who works directly with the GGA. And we're really excited to bring to you Dr. Lisa Chadwick. So Lisa got her doctorate in genetics from Case Western Reserve. And then she came down to this area to do her postdoc at NIEHS. So that's what brought her to the area. From her postdoc, she did a career pivot, I guess you could say, and became a program officer at NIEHS. So that's where I got a chance to meet Lisa. And I would say, while we worked together, one of the things that impressed me most about her is that she was very much on the cutting edge of what was coming next. So she introduced us to microbiome before any of us knew that that was a thing. One of the other, not just scientific things, but she was also very pivotal in terms of learning how to work remotely. So she was the first person at NIEHS who worked remotely. She moved to Florida. Her family moved there, but she continued to work with us at NIEHS, and this was well before the pandemic. She taught us all how to know how to use the mute buttons on our video conferencing and teleconferences. And she was a pioneer in many ways, both in terms of technology and in terms of science. And she's also wonderful at introducing people to the NIH and understanding how the NIH fits together. So I've worked with her on several different presentations to postdocs and to grantee organizations. And so I'm really excited to bring her here to tell us a little bit more about what she does at NHGRI and what NHGRI is, just to get a bigger picture. So with that,
great. Thanks. Well, I'm really excited to be here. I did used to live in Durham, so I will pretty much take any opportunity that I can get to come back to the Triangle. It is my favorite. So I was happy to be here. So I'm certainly not going to talk for a full hour because I don't think I could talk about what NHGRI does for a full hour. You would all be so bored. So there will be a lot of time for questions. I'm happy to take all manner of questions from people in that time. So, okay. So first I was going to start by introducing myself. So Molly gave a little bit of that introduction. Like she said, I have a PhD in genetics. I studied the genetic factors that influence X chromosome choice and X inactivation. And I did QTL mapping, and I spent a lot of time in the mouse room breeding mice. And my PhD advisor moved from Case Western Reserve to Duke when I was towards the end of my PhD research. So that's how I ended up in the Triangle. I finished up my degree. I did a postdoc at NIEHS in the Extramural program, which is, if you're looking for postdocs, it's a great place to do a postdoc. You're like the only trainees that they have are postdocs, basically. So you are really the focus of all of the career development activities. It's really a great thing to do. I did that for 3 years, and then I was like, okay, what's next for me in my career? And I had planned on going into academia. I think my whole career, that's where I had thought I was going to go. But I think when I got to the point where I needed to go on the job market for that, I realized I just didn't feel like I was in that sort of mindset anymore. And so I looked at other options. I applied all over the Triangle and all of the biotech companies, everything around here. And one of the benefits of being a postdoc in the intramural program at the NIH is that you see all the job postings that come through. So a job posting came up looking for a program officer to lead parts of our epigenomics program. And I had enough epigenetics experience that I was a good candidate for that. I had no idea what a program officer did, not a clue. Fortunately, my postdoc advisor did. And he was like, I think you'd be good at this. I think you should apply for it. And so I did. And I was a program officer then at NIEHS for 10 years. I was involved in leading a program called the Roadmap Epigenomics Program, which is where we developed epigenomic maps in a variety of normal human cell types and tissues and created like a public data resource that maybe you've used. I always love it when I see people talk about it in their talks. And I also led some of the NIEHS programs. So I started our program in microbiome research. At that time, the NIH had the Human Microbiome Project going on. I was like on the working group for it. I didn't know anything about the microbiome before that, but I learned a lot and started to see the connections between toxicology and the microbiome and what that could look like. And so started that program. I also had program in transgenerational inheritance, and then some of our environmental epigenetics grants. And then about 6 years ago, I moved to NHGRI, the National Human Genome Research Institute. So there I lead a program in rare disease genomics. I won't talk too much about it. The program that I lead is called GREGOR, which is like a great acronym that stands for Genomics Research to Elucidate the Genetics of Rare Disease. And I do some of our other rare disease genomics programs. I'm also the lead for our early stage investigator programs. So I do a lot of talking to early stage investigators about the NIH and how to navigate it, how to write grants better, et cetera. And then I have grants in my portfolio that are in our genomic technology development program, which is like developing all kinds of new genomic technologies. So that is why I'm here. This is like some pictures from like back when I started working in genomics. I've been working in this area since I graduated college, which was, you know, almost 30 years ago. And before I went to grad school, I worked in a lab where I was doing linkage mapping and positional cloning of rare disease genes. And so this is what it was like. We did our sequencing and genotyping with radioactivity. Like I lost many items of clothing to like the hot drawer because I got a spot on it and you had to leave it there to decay. You know, we ran everything on gels, read them off by hands, reading all the sequences by hands. I looked for a screenshot of the NCBI website from then because I used that a lot. I used BLAST a lot and I couldn't find that. The closest I could get was the 1997 logo from the NCBI News, which is looking pretty 1997. And like at that time, like you had to get your BLAST results back by email. So things have really changed a lot since I started working in genetics and genomics. The year that I started grad school is when the first draft of the human genome was published, and that was obviously a huge milestone that really changed how we do human genetics research. That first human genome sequence cost about $3 billion. And we've made huge leaps forward in sequencing technology since then. So we're getting close to like $100 to generate a human genome sequence. Now we've sequenced millions of human genomes. We have a lot of information about the normal range of variation that we never had before. We also now have technologies that have given us the ability to look at the genome and how it functions in ways that we were never able to do before. So through programs like ENCODE, which is another major NHGRI program that you might have heard of, we learned a lot about the functional elements in the genome and how they work. We've made real major advances in how we understand the genomic basis of human disease. Like I said, I work in the rare disease space and I have like from a lot of my career, and that has completely changed the way that we approach those kinds of studies. But we're also now learning a lot more about the genetics that underlie complex common disease and disease risk. And then we're starting to see genomic medicine being used in the clinic. And a lot of this work is due to research that was funded by NHGRI. So I wanted to like start by putting NHGRI into the context of the larger NIH. So the NIH budget overall was in 2024 was $47.1 billion. It's made up of 27 different institutes and centers, and each one of them has their own very specific research focus. Most of the NIH budget, over 80% of it, is awarded to researchers at institutions like this as competitive grants. And each year the NIH supports over 300,000 researchers in every US state, many other countries in the world. I think it's about 2,500 different institutions receive NIH support. And then about 10% of the NIH budget supports research on the NIH campus in the intramural program, like when I was a postdoc. So NHGRI is one of the sort of smaller, mid-sized to smaller institutes. It's the one with the orange arrow. So I just wanted to give you sort of an idea of what the size of NHGRI's budget is relative to the budgets of the other institutes. No one comes close to NCI, they're enormous, right? Okay, so this is a figure that came from our 2020 strategic vision document, which was published in Nature a few years ago, and I'll talk about that later in the talk. The top graph shows how the NIH budget has changed since 1990. So the NIH budget overall has increased a lot since 1990, thankfully. And the bottom graph shows the NHGRI budget in those same time periods. So it's also grown over the years, and the scale of these is not the same. If they were on the same scale, the part that is NHGRI would be so small. So I made another sort of scale thing. The NHGRI is like basically like one finger, right, of the NIH. So our budget is comparatively very small compared to what else is at the NIH. And then it's also, I think, surprising to people that like another thing that has changed over time is how much of the genomics research that's funded by the NIH is supported by NHGRI. So during the Human Genome Project, which is that first set of circles, almost all of the research that was counted as human genomics research at the NIH was supported by NHGRI, and that was mostly the Human Genome Project. Even in 2010, about 50% of the human genomics research funded by the NIH was supported by NHGRI. And this is 2020 data, and I would imagine it's similar or even getting lower, right? That we actually only support 10% of the human genomics research at the NIH. So the tools and methods that are developed by researchers that are funded by NHGRI are being used to understand the biology of a lot of different, processes, diseases, and everything. But we don't support that kind of stuff. We support more of the foundational stuff. All of the institutes have a very specific mission that guides sort of what kinds of research we accept and don't accept. So this is NHGRI's mission. It's a very lofty sounding statement. I won't read it. But we really focus on the tools and the technologies that help support using genomics in the broader context of human health and disease. And the forefront of genomics is like our, what they call it, motto or something, but we use that a lot. Okay. So I talked to Molly before this talk, and I was like, a lot of times I give these grant writing talks, and I haven't really given a talk like this before. So I said, well, what should I talk about? She said, well, people don't always understand what NHGRI does and doesn't support. So I thought I would talk about some of the NHGRI rumors that I've heard over the years. So the first one that I hear a lot is that NHGRI doesn't take R01s. So I assure you that we do take R01s. So these two graphs are data that I pulled from NIH Reporter, which if you don't know how to use NIH Reporter, you should go learn. It's a database of all of the funded research by the NIH. You can get so much information about who funds what, who's doing what study sections it gets assigned to, who the program officers are, tons of information in Reporter. And the top graph shows every fiscal year the amount of money that we put into new or competing renewals of R01s, R21s, R03s, and R35. I group those all together. And so we do support a large number of those grants every year. The graph at the bottom shows what we fund every year in new and competing renewals of cooperative agreements. And that's what we use for like the consortium type work that we do. Cooperative agreements are grants where the NIH staff have like a much more involved role in directing what you're doing. And we do use a lot of those, but I did want to show that comparatively every year we are actually awarding more R grants than we are U grants. I mean, except in 2021 when we had a number of large programs start that year. That's why that number is so big. But again, we really are known for team science and the kind of work that we do in consortia. This is just highlighting some of NHGRI's funded programs and projects. or programs that are funded through the NIH Common Fund, which if you're not familiar with what that is, it's like kind of another, it's not really an institute, but it has its own budget and it funds transformative research that really doesn't fit in the boundaries of one specific NIH institute. A lot of times it's like resource development or technology development. And then when those programs end, that kind of work goes into the other institutes. So the Human Microbiome Project is one example of an NIH Common Fund program. So is the Roadmap Epigenomics program that I was part of. If we put all the other sort of international groups that we work with, you wouldn't even be able to read the slide. It would just be like a ton of fancy logos. So we do a lot of that kind of work, but we do support research grants as well. Okay, so this is another one that I hear. I heard NHGRI doesn't fund model organism research. And that is certainly not true either. In fact, we are the lead institute for COMP, the Knockout Mouse project. We also support the model organism databases and the Alliance of Genome Resources and things like that. So we do actually do a lot with model organisms. And I think to think about sort of what NHGRI funds and what they don't fund, it isn't really, that's not sort of the criteria. So we'll talk about some examples of things that we do or do not fund. So this is some language that I took from a funding announcement that we participate in where we had to put sort of language about what we take. And I put squares around a couple of the things that are really important that to understand what we support. So we do sort of that research, resource, technology development, methods development for things that are generalizable. So we don't support something where you're developing a new method to look at one specific thing that's only going to really work in that context. We develop methods where you could apply it to a bunch of different diseases or genome-wide or, you know, any sort of biological process. And that's more of an NHGRI kind of thing. We don't fund things that are like what I call using genomics tools to study a disease or a biological process. So like, that's the kind of stuff that is funded by the other institutes of the NIH. They're all interested in using those kinds of tools, but we're not really the ones that fund sort of what is the molecular basis of a specific disease. We fund how do you figure out what the molecular basis is of a disease. So that's kind of how to think about what is NHGRI and what's not. Also, if you're not sure and you're trying to figure out, you know, where to send where your grant application is going to go, you should talk to a program officer. And I made that. Yes, I love this. This is the old school field guide. Yeah, so that's like one of the big things that I hope you take from the talk is how important it is to reach out to a program officer before you submit your application. So you can send me some aims and I'll look at them and I'll tell you whether they're sort of in scope for NHGRI or whether there's a better home for them somewhere else. And I can explain why. If you're applying to a specific funding announcement, I can tell you whether this is the right thing for that or whether you'd be better off applying through some other route. I listen to a lot of grant reviews and I know sort of the personality of the review panels and what they're looking for. So if I read your aims and I know your application is probably going to go to our study section called GCAP, which is where a lot of NHGRI's applications go. And I see that you don't have like a benchmarking part of your technology development grant, I'm going to tell you to make sure that you include that because that's something that I know that they are going to want to see. And then we can also help just sort of look over your aims and see if there's any sort of major mistakes that you've made. So you should definitely reach out to a program officer before you submit, after you get your summary statement back. Every time there's an opportunity to do so, it's a great thing to do. I also wanted to talk about how you interact with a program officer to have that be like a beneficial interaction for both of us. So one like major thing is like don't just call me. First of all, I don't actually know that my phone number is hooked up to anything anymore. It's like, somehow, if you call, like, I sometimes get messages, but like, I'm not even gonna answer that because I don't know what you're calling me to talk about. What am I gonna tell you that's gonna be useful? I haven't seen your summary statement. I don't know. I haven't read the aims that you're thinking about. It's better if you send that ahead of time and I've had a chance to think about it. So do send an email to the program officer. And then when they don't write back to you, send them another email. And then when they don't write back to you again, like send them another email. Just like all of you, we get so much email and it's so hard to keep track. And if I don't respond to you, it's not because I'm not interested in helping. It's because I have gotten like 8,000 emails since you emailed me. And that email is so far down the inbox that I might never see it again. So don't hesitate to do that. When you send an email to the program officer, before you submit, it's a good idea to include the draft specific aims in that initial email, because I can tell you then whether this is even the right fit for us or if it's not. And if it's not, there's no point in us having a call. Like, I'll just tell you who the right person is to talk to. So that saves a lot of time for everyone. And then set up a time with me to have the call or have a Zoom. I mean, actually, that's one thing I really like about the fact that everyone is using Zoom now. I get to have face-to-face calls with my PIs. It's great when you're talking to them. I was telling them at lunch, it's like less great when I'm giving you bad funding news, which I do that too. But in general, like, that's how you should interact with your program officer. And then I thought I would go through a couple of like examples of things that like would be appropriate for NHGRI or would not be appropriate for NHGRI. So like on the left are things, examples of things that I think are appropriate for NHGRI. So you're developing a new large-scale technology that looks at some aspect of the genome or how it functions and does it on like a genome-wide level, not just at like 5 genes or not just at like centromeres or not just at like whatever, like a kind of a large scale. Or computational tools, like developing computational tools that integrate a bunch of different molecular data types to help I put identifying prioritized candidate genes and variants in individuals with rare disease, because that's the kind of thing that I care about. But it's like developing tools that help you do something that can be used in a whole bunch of diseases. It's not like it's only going to work in like cancer. It's only going to work in immune diseases. But the examples of things that are not within scope for NHGRI are things like using those large-scale genomic technologies to investigate the molecular basis of a disease or a molecular process. That is something that the institute that funds the kind of research in that area would fund, or using sequencing and other molecular analyses in a cohort of individuals with disease to try to identify the gene that causes that disease. That's also not something that we support. We would support ways to do that more effectively, but not like trying to identify the gene that causes a specific disease. And the other thing that I wanted to point out is like, it's also really important how you frame things. So I talk to people sometimes who send me a set of specific aims and I'm like, well, okay, I kind of think that like what you're doing could be NHGRI, but you need to change the way that you frame it. So an example would be if somebody sends me some aims and basically it starts out like, I'm interested in understanding the genetic and environmental basis of huge toe disease, which is ravaging our country, right? And I know that huge toe disease is caused by a mix of genetic and environmental factors. And I'm going to develop a new analytical method that's going to help me identify the genetic and environmental contributors to huge toe disease. And like, once we do that, that's going to help us develop therapies for huge toe disease. So like, that's a really interesting project for the Huge Toe Disease Institute. It's not really an NHGRI project, but you could take a lot of that and turn that into something that would be relevant to us. So you could rewrite that same thing, like, many diseases are caused by genetic and environmental factors, but it's hard to figure out what those are. And we need new analytical methods that can help us identify both of those and how they contribute to complex disease. So I'm going to develop a method like that, and I need to test it to show that it works the way that I think it is. And huge toe disease is a great example to test it in. So I'm going to use that to validate my method. But you could use this to identify the genetic and environmental causes of, huge thumb disease too, or other diseases of hugeness on your body. So, so that's sort of an example of how to think about the kind of work that you're doing and how you can spin it to be a better fit for NHGRI, if that's something that you're trying to do. So if you are interested in submitting grants to NHGRI, like I said, you want to talk to a program officer. There's a lot of ways that you can find the right program officer to talk to. We have a page that you can navigate to through the research funding part at the top of the page there that has scientific contacts by research area. And so if you go there, there's like a menu. You probably can't read this because it's small, but like it breaks down all these different sort of major categories of research. And if you click on the arrows in each of those, it'll expand it to some subcategories. And then if you click on those subcategories, it'll take you to a page where the program officers who have those kinds of grants in their portfolio are. So that'll give you an idea of like who the people are for you to talk to when you're thinking about submitting a grant. Okay, so the rest of my talk, I'm going to talk about sort of where NHGRI is going and how we figured that out. So even though I told you at the beginning of the talk that like we've made so much progress in genomics over the last 30 years, we still have a lot of challenges, and some of them are listed here. So it's still really hard to use genome sequence data in a clinical setting. In genomic studies, we have like an overall lack of diversity in the data that we have, and that causes a lot of problems. I mean, it's hard to interpret variants because you don't know whether a variant is like normal in a population or not normal in a population. So that's a problem. We also still don't have a great understanding of how the genome functions in different sort of cellular and spatial contexts. And then, of course, we all know that, I think the genome's really important, but it's not the only thing, right? We know that there are a lot more than just the genome that contributes to health and disease. And so NIH institutes like periodically go through these sort of strategic planning processes. It's so fun. It involves a lot of meetings. So we had our last strategic plan came out in 2020. This is sort of like how you develop a strategic plan. We had a lot of meetings where we had people from the community. They came in, they talked about the things that they thought were the biggest challenges, where they thought the opportunities were, what their ideas were. Somehow that all got turned into the strategic vision document, which was published in 2020. And then now we're in sort of the implementation phase of that, where we're taking those major priority areas and we're turning them into programs that we fund. And so I'll tell you about some of the parts of the strategic vision and some of the programs that we fund that go along with those different parts. So we got this really nice graphic that illustrates the 4 major parts of the strategic vision. I'll go through each one of these. I think if you're interested in learning more about our strategic vision in general, I would encourage you to just read it. I'm in the Division of Genome Sciences, so I have a very genome sciences view of the things in the strategic vision that were important to me. But we also have the Division of Genomics Medicine, and we have the Division of Genomics and Society. So there are other aspects of the strategic vision that I might not talk about here, but maybe of interest to you, so you should read it. Okay, so the first sort of major section is the guiding principles. And so I listed some of them here. Like one of them is that, we want to strive for diversity in all aspects of genomics research, and not just in the individuals that we have sequences from and that we include in those studies, but also in the workforce of people that do genomics research. Actually, genomics has been we all know that there's a problem with diversity in the workforce in science and research in general. Genomics is like the worst. I remember a friend of mine was at a talk sometime and somebody was talking about a paper where they had looked at a bunch of different fields and genomics was highlighted as the absolute least diverse of any of them. So we have really been working on that. We also want to make genomics as useful and accessible as it can be to everyone, not just researchers. We want to consider how genomics in combination with social and environmental factors contribute to health and disease. We want to promote the use of standards for how we generate, analyze, store, and distribute data, because we generate a lot of data that can be useful. But if you tried to use other people's data, which I'm sure that many of you have, you know that without standards for that, it's like not easy, right? So we do a lot of like standard sort of development. And then we are also really committed to open science, data sharing, and collaboration. Those are things that have been important to NHGRI for probably since it started. I think genomics research was one of the sort of early areas where we really saw a strong push towards data sharing. And so that's really just part of our culture now. So some of the programs that I wanted to highlight that fall into this category are some of our efforts towards increasing diversity in the genomics workforce. So we think about that from sort of all levels of the workforce, from college to even high school, and how do you get high school students from a range of backgrounds interested in genomics, to people who work in like a clinical lab, like doing, you know, blood testing or whatever they're doing. They're doing some of the genetic testing in there, but they don't know a lot about what they're doing. So we have like courses and stuff that are aimed at that kind of group. So we try to focus on the whole pipeline. We have an office that coordinates most of this kind of work. It's called the Training, Diversity, and Health Equity, or TIDE. I didn't put TIDE here, but that's what the acronym is, office. And this is just a figure from their sort of strategic plan document. Some of the current programs that I thought I'd highlight were these three. So we have a program called Diversity Centers for Genome Research. That is sort of helping to build the infrastructure and provide opportunities for research and training in genomics at minority serving institutions. It's really hard to like start to get funded to do genomics research when you don't have the infrastructure to do genomics research on a large scale. And so this sort of helps diversify the kinds of institutions that are able to do that kind of work. The GREAT program, I listed the RFA number. That RFA has expired, but I think it's being renewed. So that is focused more on undergraduates and getting undergraduates from diverse backgrounds into research genomics careers by providing research experiences and training. And then we also support a lot of work in computational genomics and data science. We have another program called the Educational Hub and Partner Sites, and our for computational genomics and data science. Our hub is actually at North Carolina A&T, and they provide sort of those educational and research opportunities, but in the computational genomics space at institutions that are not well represented in this field so far. So that's some of those current programs. So the next part of the strategic vision is a robust foundation. So we want to do things like enable the routine generation and analysis of increasingly complex genomic data, use evolutionary and comparative genomics to help us better understand genome function, develop methods and sustainable resources for genomic data sharing and computing. That's, of course, like so important. Just sort of overall increase genomic literacy across all sorts of aspects of society. And ensure that genomics researchers and healthcare providers can use genomic data to their fullest extent. So some of the things that I wanted to highlight here that are more relevant to the Division of Genome Sciences are our genome technology program. So that is how we fund all the technology development research. the goal there is to accelerate innovation, development, and dissemination. That's a really important part of that, of genomic technologies. We have a whole bunch of different ways that you can have a grant that comes into our genome technology program through specific RFAs, like we had for a number of years, RFAs for sequencing technology development. So funding research into developing new ways of doing sequencing, hopefully to continue to get that cost down. We take R01s and R21s through the parent announcements that are doing genomic technology development. We have a NOSI that, I hate saying NOSI, it stands for Notice of Special Interest. It's basically like an institute telling you, here's an area of science that we also like and we want to get applications in. We don't have any money for it in particular, but like we like it. So write that. So we have that. We have had funding announcements that were for developing new ways of doing single molecule protein sequencing. We also have, all of the institutes have funding that they give for small business grants. And so we have a lot of technology development that happens in the small business space. And then we've had RFAs for synthetic nucleic acid development. And then we have a coordinating center that actually coordinates all of our genome technology grantees. So they have an annual meeting, they bring them all together to talk about what they're doing, learn from each other, network. They have recurring seminar series and stuff. So it's really just a way to connect our technology development community. And then we also have the Centers of Excellence in Genomic Science. That's a larger grant program that funds sort of really innovative, transdisciplinary kind of work in genomics, developing new technologies, having really innovative ways of getting them out to users and applying them to problems and sort of showing how they can be used. So those are some of our technology development announcements. Another thing that I wanted to highlight that I think falls into this category of a robust foundation is the Genome Reference Program. So when you align your sequences to a genome reference, the Genome Reference Program is funded by NHGRI. Right now, what they've been working on is developing what are called the T to T genome. So a gapless genome that doesn't skip all the hard parts. And then turning that into what they call the pan-genome reference. So that is to address the limitations in the current most frequently used genome reference, which is H38, which has gaps and also has no haplotype information, has no information about variation. And so what they're doing in the pan-genome project is they're sequencing as many sort of genomes as they can from a range of genetic backgrounds, and then putting them all together into a pan-genome, and then developing these ways of working with the pan-genome. So this was the cover of Nature, and you can see all the lines and the trying the points that come up, and that's basically a pan-genome graph, right? So it shows sort of the parts of the genome that everybody's the same, and then there's a part of the genome where there's a lot of variation among the people who make up the pan-genome reference, and then it goes back. So it's just more reflective of what's actually happening in human populations. And then they're also working on developing informatics tools that enable researchers to use it. So, working in rare disease genomics, a lot of the clinical genetics labs are using HG19, which is the really old genome reference. And that's because every time you move forward to a new reference, you have to redevelop all the tools that you use. And it's a lot of work, and they don't really want to do that. So we're working on developing tools that sort of make that genome accessible and usable by a whole bunch of different research communities. Okay, so the next section is the breaking down barriers section. So some of the points that are raised in that are that we want to make it easier to look at the functional impact of genetic variants by sort of scaling up the technologies for doing genome editing. Establishing the means to determine the functional consequence of variants and how they contribute to health and disease, and understanding the role of genetic variation, normal genetic variation in disease. So I'll talk about two of the programs that I think fall into this one. This is the one I know the most about. This is GREGR, my rare disease program. And the reason that this program exists is so even when somebody we're pretty sure that someone has a genetic disease that we're pretty sure is probably caused by one major mutation or one major gene, those individuals go in, they get the standard sort of clinical genetic testing, which is the standard is whole exome sequencing. So it just sequences all of the exons and it looks for mutations. And over half of the people that get whole exome sequencing don't get an answer. We are not able to find the variant that causes their disease. And so GREGR is developing new ways to look at genome sequence to try to figure out how we can identify causal variants in individuals that don't get solved using full exome sequencing. So this program started in 2021. We have 5 research centers and a data coordination center. And they're working on sort of using whole genome sequencing instead of whole exome sequencing and other technologies like RNA sequencing, methylation analysis, things like that to see whether there are new ways to look for causal variants. One of the big things that we do in this program is we release the data to the community so that others can use it. Anvil is NHGRI's cloud computing platform. So actually a lot of the data from programs that I talk about today is on Anvil. We tried to make it as easy as possible to request and access this data by limiting the number of consent groups. I don't know if you've ever tried to use like human disease data in dbGaP, but you know, it's split into consent groups. And sometimes to get a full data set, you would need to request like, 20 different consent groups because some are only for this and some are only for this, and you have to request them all separately. So we tried to minimize that as much as we could to make it easier to request. In our upcoming data release, we have data from over 3,100 families, which includes at a minimum their sequence data. Sometimes they have things like RNA-seq. They might also have long-read whole genome sequencing and the phenotype information that goes along with them. And so that's a really valuable data resource. So, so one of the problems that we have in GREGR is like, we're pretty good at assessing the potential functional impact of variants that are located in coding regions. And we're pretty bad at figuring out what variants outside of coding regions we should look at. And we need information about that. So one of our, oh wait, I have another slide in the middle. This slide isn't that exciting. This is basically like talking about how using whole genome sequencing, which I thought at first was going to make a huge difference because now we're looking beyond the exomes, we're looking at all these other parts of the genome. And it does help us increase the diagnostic rate, but it's actually a lot less than you would hope. And so that's where I was getting to. Part of the problem is we don't really know how to look at those regions outside of the exomes to figure out what is important, what we should look at more. So we have another program. This is the much larger program called IGVF, which stands for Impact of Genomic Variation on Function. And basically what they're doing in that program is doing a systematic perturbation of the genome to look at the impact of different genetic variants on genome function and phenotype. And a lot of the phenotypes are molecular phenotypes, but they're also using things like organoid models and looking at phenotypes that are like more biologically relevant phenotypes. They look at the level of the individual variant, but also on a network and pathway level. They're doing things like developing predictive models so that you can look at a new variant that you don't have any information about and make predictions about whether it's functionally important. And then they also disseminate those findings online. They have a portal where that information is disseminated. So those are two of the programs that I think fell into that sort of breaking down barriers category. And then the last one is compelling projects. So there were more to this. Once I got to this side of the arrow, I couldn't fit that many bullet points here. So you'll have to read to hear more of the compelling projects that were called out in the strategic plan. But some of the things were to get a more comprehensive view of the roles and relationships of genes and regulatory elements and pathways and networks. And one was about extending the use of multi-omic studies in disease in taking that into a clinical setting. So the last program I'll talk about is the multi-omics of health and disease program. This launched even more recently than Gregor, so it's really kind of just getting off the ground. But what they're doing is applying multi-omic technologies in a diverse population of people with diseases, and they're trying to learn how to use that data to sort of look at how disease progression changes and how you can predict that in the molecular data. They're developing new methods to do that, new like sort of data analysis and integration methods. And then all of our programs sort of disseminate the data, so that data is also being disseminated. I'm pretty sure that this one is also on Anvil, which is where the Gregor data is. So the last thing I wanted to highlight before I left is I mentioned earlier that I also lead our early stage investigator programs. So I put together a website for early stage investigators at NHGRI. And on that website, I have links to any funding announcements that I think are relevant to early stage investigators. I have, I give like the grant rating talks, and I have video recorded a couple of them. And so I have those posted there. Those are not NHGRI specific. Those are just really like, how does the NIH work? Like, how can I do better at writing grants and navigating sort of that system? I have all kinds of other things. So I would encourage you to look at that page. I think this QR code will take you to it, but you can also search for NHGRI funding early stage investigators. Here's one of the videos that I have posted up there. And then I also wanted to highlight that we do have an RFA that is for early stage investigators, people with early stage investigator status. If you don't know what that is, you can ask me in the questions after this. That is not active right now, but I just recently presented the concept at council to renew it, so you can expect that it's going to come back. It just hasn't come out yet. So that was the last thing I was going to say, and now I'm happy to take any questions that you might have. Okay.
Do you have any tips for not being scared of program officers?
Well, we're very scary. So the main tip is like, it's like I'm a program officer, right? And I'm very representative of, I think, program officers. It's our literal job to help you. Like, that is like why we exist at the NIH. And so you should not hesitate to reach out. You should not hesitate. You know, people email me and they're like, oh, I'm sorry, I don't want to bother you. I'm like, no, that's what I'm here for. So yeah, I did talk to somebody this morning and they were like, well, not all program officers are that great. And so that's true. Like, not all of anybody is that great, right? And sometimes you're going to get somebody that's not going to be that helpful. But in general, we go into a job like this because we like to help people. And so you shouldn't hesitate to reach out to program officers. We're like just normal people. Go ahead.
Wondering, integrating different sorts of data with genomic data, are there any sort of high-level lessons that have been learned? It seems like an incredible challenge.
Yeah.
For a lot of people, yeah.
I would say the high-level lesson is it's hard. No. And I think like there's a lot of reasons why it's hard that are hard to solve. So one, to me, seems that the kinds of people who are doing each one of those things are not the same kinds of people, and they don't always communicate well with each other. So I remember when I was at NIEHS, we had people who were interested in doing environmental epigenetics, and they wanted to use like all these like fancy genomics techniques to look at how environmental exposures impacted health or gene expression or whatever. But like they didn't really have the great, really strong expertise in doing that kind of work. And then we have genomics researchers who say like, well, we're gonna show how environmental exposures affect this. And they're like really good at doing the epigenomic part of it. And they don't know anything about doing an exposure study. And so they're doing something that's not really like relevant. It's not really a relevant exposure. It's not the right kind of system. They're not looking at the right phenotype. They're not recording the data in the way that's, so partly I think that just building those communication pathways is really important. And like developing standardized ways for talking about the data. And then, yeah, once you can just communicate with each other, then we need people to develop the tools. But you're right, it is a hard challenge for sure.
So we started to talk about this a little bit in our meeting earlier, but I wanted to know if you could comment anything about NH thinking about rising costs of people doing research and what the vision for that is going forward.
Yeah, so I know that that's a huge issue that we're all facing because everyone deserves to get paid like an amount of money that is reflective of their expertise and the work that they're doing. The flip side of that is that the NIH budget is only what we get, right? And it doesn't go up as much as we would like. A lot of times when it goes up, it's being earmarked for specific things. We can't even use it to necessarily do what we want to do. We have to use it to fund this program that some senator liked, right? And so it is a really hard problem. And I think to solve it, it's going to take institutions and the NIH both working together because neither can solve it themselves. We are, I know that things like our stipend levels go up, they don't go up enough. But the flip side is like the more money you put into that, the fewer people you can fund. And so it's such a hard balance to do. I know a number of you that I've talked to today have like grants from NIGMS. And NIGMS has one very specific funding model, which is like, we're gonna give money to as many people basically as we can. You don't get a lot of money from us, but we're gonna give it to a lot of people. And it's really a hard sort of thing to balance. But I think it's gonna take both the NIH and institutions.
Hi, so one thing I was thinking about as you've had a lot of experience in this, how has the types of grants that get funded changed since like you began? So like what types of things in a grant help it get funded nowadays compared to like when you just started out on this job?
I don't know that I would say that I can point to anything in particular. I was talking to somebody earlier about some of the best tips that I have though for people and what are the things that I think help a grant get funded. So one of the biggest tips that I tell everybody is just sort of understanding who you're writing for. Unless you're planning to fund your research yourself, you are writing for reviewers, right? And so you need to know sort of who they are, what they're looking for, and how to give it to them in a way that is digestible. And when I talk to people, a lot of times they're like, well, reviewers are scientists that are very close to my field and they understand what I'm doing and they already know. Well, that's not true. So they actually aren't, right? A lot of times reviewers are people that are sort of kind of related fields, like they sort of generally understand what you're doing, but they're not exactly the same kind of scientists as you. And so if you write something that is only understandable by people with your specific expertise and only people that care about what you care about are going to understand why that's important, then you're not going to get funded, right? You need to like make it really obvious to like your mother, right? Why what you're doing is important, why the problem needs to be solved, what the impact of solving that problem is going to be, and not write that in a way that's not understandable. The other thing that I always tell people is like, the people who are reviewing your grants are people like you. They like go to work, they run their lab, they write their own grants, they teach their classes, then they go home, they like feed their children and put their children to bed. Then now it's like 10 o'clock and they're sitting down to read your grant. and like 10 other grants, right? And like, think about what you want to read at 10 o'clock at night. And what you don't want to read at night is like, open up a document with like a page just chock full of words and like, very hard to understand. If that's what you're giving people, they don't even get past the first page before they're bored, right? And there's no real incentive for them to keep reading. So I think it's more about the way that you present your ideas than anything else.
Do you have any predictions for where the field is going in the next 5-10 years?
So we had a whole seminar series that you can find actually on NHGRI's website, bold predictions in genomic science. I can't even remember what they were all about. The one that I remember the most was about really like taking genomic information into clinical genetics and in the clinic and really getting that integrated in a way that it isn't right now. But you might be interested in reading and watching those videos. I think there's like 5 different seminars where 2 people got together and talked about some of their bold predictions in some of those different areas.
Thanks.
So, first of all, I'm asking this question on behalf of myself, not on behalf of NC State. So as you might know, North Carolina is one of the states where some recent legislative actions have changed our ability to offer programming or training related to diversity, equity, and inclusion, or, you know, in some cases to even collect demographic information about our students. And particularly in thinking about how we write training grants, I know all of these things are incredibly important. And we've been advised and guidance we've received from the state and the UNC system that we cannot include these things in our grants and that we should just explain to our funders that we are not allowed to do so. And so I'm sort of wondering what guidance or suggestions you can make to us in terms of us being able to successfully write these grants.
Yeah.
And in terms of what we could better explain to the folks making these policies.
Yeah. So I live in Florida, so I understand. So we were talking about this at lunch actually, and like I actually myself personally don't have a lot of advice to give you because I haven't had grants yet that have had like a PEDP as a requirement. I haven't really had a lot of my own experience in seeing how those are talked about and reviewed. But I understand that that is coming to more and more grants. Now, the other thing we were talking about at lunch is that we get the same sort of thing though from the top too. So it's changing all the time from all aspects. So an example is, you know, we provide supplements for the diversity supplements, and they're to bring in people from diverse backgrounds into science, right? And even like the language that we are able to use and what we say is a person that is a good applicant for a diversity supplement has changed in a way that makes it almost like not really a diversity supplement. So I get that. I think the thing that I would say is to talk to, at every institute, there's going to be for training grants, like a person or a couple people who are the program staff that work with those. They are the ones who hear the reviews and sort of know what reviewers say are looking for and things. And they can probably give you some advice about how to sort of thread the needle of like conveying what you are really thinking without saying it. I think that that's happening in a lot of states, and it is something that we're going to be having to grapple with. But we're also only not that long including plans like this as part of research applications. So like everyone is sort of getting used to it. So that's my best suggestion, is to talk to the program staff who do training grants. They'll probably have better advice for you than I will. Okay.
for.